Hey guys, so today we're going to talk about cells, and I just wanted to show you this picture of a bunch of different types of cells. So this over here are nose cells, these are onion cells, and these are a type of bacteria cell. And all of these are cells that we can see using various types of microscopes. So a microscope is an instrument that magnifies an object. Now, we are going to use this type of microscope here. Ours has just a singular eyepiece, and it has 3 objectives, and the objective is how close you're going to get to the slide, and so how large it's going to magnify. This stage is where you put the slide, and you can move the slide up and down. Coarse focus brings things, it's like if it's really out of focus, it will bring it into focus. Fine focus is more detailed. And then here is your light source. This is a dissecting microscope. We don't have these here, but these are basically just used to, like, if we were to be inspecting flies, for instance, we could take a fly and magnify it and look at all the different parts of a fly. Now, sometimes these particular microscopes are not super great, or they don't have enough magnification to get to some things like viruses and bacteria. So here is Salmonella bacteria on the left-hand side of your screen using the light microscope like we would have at school. This is using something called a scanning electron microscope or micrograph. And this is basically an electron microscope, and it uses beams of electrons, which we know are negatively charged particles, instead of light. And it allows for a higher magnification and more detail. So here is a picture of the difference between a light microscope and an electron microscope. You can see this electron microscope is quite large. So typically they only have these in university type settings. So what are we looking at under the microscope? Well, we're going to be looking at cells, and we know that cells are the smallest unit of life. And we know that because of the cell theory. This is the first unifying theory of biology. Cells are the fundamental units of life. All organisms are composed of cells, and all cells come from pre-existing cells. And all of that sounds like, duh, we already know. But back in its day, it was revolutionary. Especially the cells come from pre-existing cells. Basically, life has to come from life. Studying cell biology is the same as studying life. So if you're looking to study anything living, you really should be looking at their cells. Now we have 2 main types of cells, prokaryotic and eukaryotic. And karyo, K-A-R-Y-O, means nucleus. So this is before pro-nucleus, and eu meaning true nucleus. Okay, so prokaryotes are without membrane closed compartments, which we're going to call organelles. They don't have a nucleus, whereas eukaryotic cells have organelles or membrane enclosed compartments, and they have a nucleus. So prokaryotic cells do have genetic information, but it's just not centrally located in a nucleus. It just sort of hangs out together in a nucleoid region, but it doesn't have anything like closing it in. So it really could float just kind of anywhere. They have cytoplasm, which inside the cytoplasm is like this water, it's more like jello-like material, and floating around in the jello are these ribosomes, and ribosomes are sites of protein synthesis, and that is going to be super important later. So this just shows you the difference. This is like a sketch, and then this is what it looks like under the microscope. Most prokaryotic cells have cell walls because prokaryotic cells are out on their own, so they're unicellular organisms, uni meaning one. So they are one-celled organisms, so they're just out in the world, and so they need a little bit of extra protection. So outside of the membrane, there's going to be some kind of cell wall. if you're a bacteria, that cell wall is going to contain something called peptidoglycans, which is basically proteins with some sugar in there. Then some bacteria are even going to have an extra membrane. So you have a plasma membrane, which is like just a normal cell membrane. Then you have the cell wall, which is like rigid and is going to provide some stability and some security. And then you even have an extra layer of polysaccharides, which we know are long strings of sugar, that is called a capsule. So this figure basically just shows you this prokaryotic cell. You have your cell wall, well, your cell membrane is this kind of darker green, then cell wall right here, then your capsule, which is thick right here. Your DNA is just floating about. Your ribosomes are just little dots floating in the cytoplasm. And then they have these things called pili, which just project out to the side, and they're going to use that to exchange genetic information. The flagella is going to help them move, or they have like little hairs on the outside called cilia. Now, cells, individual cells can only get so big, because the larger they get, the higher the surface area to volume ratio gets, or the closer it gets together. The smaller it is, the better it is. So the volume of the cell determines how much metabolic activity it has going on, so how much energy it needs. And the surface area determines the number of things it can leave and go out. So if I'm a small cell, I have small metabolic activity, and I have small things that need to come in and out, that's good. If I'm really, really big, I have a lot of metabolic activity, but not enough cell surface to get energy in and out. So at some point, the cell will either have to divide and become two separate organisms, or it will have to form a multicellular organism. So that's kind of the point of the surface area to volume ratio. And this is just shows the comparison in size. This is nanometers, so an atom. Then lipids and proteins are larger than lipids. Then you have your bacteria. The mitochondria, which is an organelle of an animal cell, is actually larger than most bacteria. So this just gives you an idea of the progression of cells.